previous speaker for thanking the organizer for giving a chance to present our most recent discovery, which I consider very exciting and offer some new insight about glycan biosynthesis. First of all, I want to acknowledge my former postdoc, Ali and Muhammad Ali, the boxing champ. Armand Petrosian in particular, Armand Petrosian is really the one behind my lab for moving from glycobiology into cell biology. Currently, he's an assistant professor. Also, I acknowledge my funding source with the VA Mary Award, NIH R21, and LB 506 from Nebraska. This is outline. I'm going to give you structure and function of mucin glycan, and then the kind of thing that regulate glycan biosynthesis. I will give you two examples. One is how does Golgi retention affect the glycosyltransferase activity, and also how the alter Golgi targeting can affect the glycan structure. And then I'll tell you what we are up to now. Just to give you some of the structure I will touch upon today. There are two types of mucin. One is secreted mucin and membrane-bound mucin. There are certain structures that are associated with secreted mucin, and they are synthesized by different type of glycosyltransferases. And there are a couple of concepts I want to introduce here. One is terminal sugar glycosylation takes place. It occurs at one sugar at a time. It's not temperature driven. Therefore, that's where the complexity comes in. And then you have up to 20 isoforms of peptidogenic transferase. There's only one single copy of the gene for this guy and this guy. And the rest of them, really multiple isozymes can involve in the synthesis of a particular structure. So the challenge is, how do you know which glycosyltransferase work on which glycoprotein? In fact, we have published 3 papers addressing that issue, which I'm not going to mention anything today. So the other concept is, when you make the CoA enzyme, there are two, maybe more than two. That's one is can make sialyl T, one can make CoA2, and then from CoA2 you can go on to make subsequent structure. And another thing I want to mention is, in a few minutes, that actually I will mention that when I get to that point. So just a few words about the function. The function, depending upon whether it's a secreted mucin or membrane-bound mucin, they have a totally different function. For secreted mucin, their function is protection. In order for them to carry out those functions, you have to have very high carbohydrate content. For the secreted mucin can go up to 80-90% of the molecule of carbohydrate. And also, they are very heterogeneous, so that they can bind to as many pathogens as possible. Therefore, their structure is very heterogeneous. In carbohydrate isolated from a secreted mucin from one single individual can be 300, 400 different structures. So that's how they protect our cell from infection. How about the membrane-bound mucin? Their function primarily is for signaling transduction. They transmit from the surface. They can guide the leukocyte migration, and also for cancer cell, they mimic this trafficking signal and to help them metastasize. And they also have high carbohydrate content, but usually about 60%. They can also help retain water. Now, in terms of the membrane-bound mucin, I'm just giving you, this concept. This is the bloodstream, this is the endothelium, this is a magnification of the interface. So in response to infection, those leukocytes, circulating leukocytes, they will be slowly tethered to the endothelium, and then they roll and follow activation and result in a firm adhesion. And they are mediated by CoA2 associated carbohydrate selectin ligand, sialyl X or sulfated sialyl X. In fact, later on I'm going to use some experimental data to show those 3 steps. Okay, about glycosyltransferases, usually we all concerned about what is a sugar donor, what is an acceptor, and other factors that can regulate it. Usually we say, okay, what is the substrate specificity that kind of motivate biochemists trying to characterize all this? Well, do you have any sugar donor? You have a acceptor, pH, so on and so forth, or gene expression. That's all people are thinking about, okay? What I'm trying to offer you today is there's another level of the in vivo regulation of glycosylation. I would introduce you about the Golgi targeting and Golgi retention that can play a very important role for in vivo glycosylation. Glycosyltransferases are made in the ER and transported to the Golgi, cis Golgi, and then retained in the Golgi according to which step they participate in, and then they recycle. Those are the papers we published recently to dealing with the first step, targeting, retention, and recycling step. And what I'm going to touch upon is this retention. I'll give you one example, even though we have published 2 papers, 2 different targets. But again, I will tell you about what the targeting side. And since step 1 and 3 are kind of linked, and the targeting can affect the glycosylation, and the recycling step, actually, normal recycling step is for recycling. But under stress condition, this step actually causes Golgi fragmentation. Just give you one concept about the in vivo glycosylation. The enzymes are distributed in the Golgi apparatus according to the step they participate in. For the enzymes that make the structure near the releasing end, they are in the cis Golgi. And then those generate peripheral sugar are located in the trans Golgi. In other words, they are stationary. But again, there's a lot of argument. If you want, I can provide you some of my insight in that regard from the glycobiology viewpoint. But anyway, so the enzymes are stationary, and then the substrate, the cargo, will come through this step. And then, depending whether they come into contact with glycosyltransferase or not, then they will be selectively glycosylated. And I gave you an example about secreted mucin. They come into contact with CoA3 synthase, CoA2M. They don't come, and this membrane-bound mucin, they don't come here, getting in contact with CoA2M and CoA3 synthase. Therefore, they don't make CoA3 and CoA4 structure. The other thing I want to mention here is the Golgi localization. This is well-established localization in CoA2L and ST3-GAL1. Actually, CoA2L localized in the cis media. In other words, when you have CoA2Ls produced, they will make CoA2, and then very little of cellular T. Okay? So just keep that in mind. That will come into handy when I try to interpret some results. Okay. So this is a retention concept and how we identify the retention protein. This is glycosyltransferase, a type 2 membrane protein, has the only thing that is exposed outside in the cytosol is the tail. And the tail can do several different things. One is they can direct from ER to Golgi transport, and Golgi targeting, and Golgi retention. And also can mediate the Golgi recycling. And basically, we use it, take advantage of this property by using the biotineated tail or immunoprecipitation, and then followed by proteomics and so on and so forth. I won't give you those data. So we identified GO3 is the one that helped retain this enzyme in the Golgi. As shown here, if we knock down GO3, this is control. The enzyme is outside of the Golgi. Okay? And this is the functional testing. I told you I'm going to show you the 3 steps. This is what it is. The tethering, rolling, and then adhesion. So this is the rolling step. E-selectin. This is the tethering, rolling, and tethering, rolling to the P-selectin, E-selectin. And either knock down CoA1 or GO3. They all decrease the tethering and rolling. And then the third step need to be activated by E-selectin. So you have to treat the cell with E-selectin and then measure the third step. Actually, the third step is more, mediated by the phosphorylation of SYK. And then you do this analysis under dynamic flow. And you immobilize the selectin and pump the cells through and measuring those properties. In other words, the Go3 is required for Golgi retention of the enzyme. Without that, enzyme cannot be in the Golgi, therefore they cannot make terminal sugar. And as diagram here, enzyme Go3 is required for CO2 retention in the Golgi, and then which in turn make PS01, CO2 associated site Lewis X, and then you bind to the circulating leukocyte and tethering, rolling, and activate SYK, which in turn activate integrin, allows this to bind to ICAM1. So now, now I'm going to tell you about the targeting issue. The study was really motivated by this publication 2007 by a group in UCLA, which they show that LNCAP is a sort of benign prostate cancer cell. This is an aggressive prostate cancer cell, where they show that LNCAP can make CO2-associated polylactosamine, and the cell can be induced to undergo apoptosis by galactin-1. And then we found that PC3 E145 can also have CO2L expression. The puzzle was why they don't make CO2 polylactosamine. So that was really a motivation for us. We're trying to find out what's going on. So we did confocal analysis on the Golgi. You can see the LNCAP cell, actually, Golgi is more compact. PC3 is more fragmented. D145 even more so. And then we analyzed where is the Golgi localization. LNCAP cell, all three enzymes are in the Golgi. PC3 cell, D145, both enzymes are in the Golgi, even though Golgi are fragmented. CO2L, actually, they are segregated from the Golgi, which means they are not in the Golgi, okay? So, and the important thing we need to know whether really those cell lines really represent the real thing. So we analyze in the tumor different degree of malignancy, and you can see that CO1G, and then ST3-GAL1, they all associate with the Golgi, even though they are fragmented. The more aggressiveness, the more fragmentation. And CO2L actually is outside of the Golgi, even in the stage 2. So what I mean is, whatever we saw in the cell line really, shown in the tumor. Okay, and way back we analyzed, we identified targeting site CO1, CO2M, this guy, using the GM130, graph 65, and GM130 giantin, and then CO2M using giantin exclusively. And then I would just go over forward to then we identify that ST3-GAL1 using GM130 and giantin, CO2L like CO2M using giantin exclusively. Therefore, in the cancer cell, when this giantin is defective, CO2L, CO2M cannot go to the Golgi, but other enzymes can still go to the Golgi. So, the other thing is we can recover the Golgi fragmentation by inhibiting non-muscle myosin 2A, it will come back. And now here's the consequence of that. In the aggressive cancer cell, they make alpha-2,3 sialic acid, sialyl T. However, they don't make polylactosamine. When we inhibit or recover the Golgi morphology, and they start to make polylactosamine, which means we also check, see what the susceptibility to galactin-1. This is LNCAP cell treated with galactin-1 causes Golgi fragmentation, and before we recover the Golgi, it's resistant. Once we recover the Golgi, it's becoming susceptible to galactose-1-induced apoptosis. So in conclusion, one, the enzymatic function of a Golgi glycosyltransferase is regulated by its retention protein. Number two, the enzymatic function of CoL2 GNT, only this group of enzymes. It happened that this group of enzymes is the focus of my whole life scientific study. That's why we found it. And then they're regulated by giantin, and because this is exclusive Golgi targeting site. So these groups of enzymes are very sensitive to the giantin functionality. So currently this is the focus of our group. We're trying to study the effect of altered Golgi targeting of glycosyltransferase induced by stress. By that, I mean heat shock, alcohol abuse, cancer by nicotine transformation, and so on and so forth, on the ending of glycan. Thank you.
Please thank Dr. Chang. Questions, please.
Very nice study, Piman. My question here is with regard to the highly metabolic nature of the cancer cells that you're working with. The Golgi glycosylation in this whole paradigm that you're talking about, is that all regulated by these receptors on the cell surface? Because cancer cell feeds from the blood in order to become metabolic active. Is this constitutively active process going on all the time in the cancer or not?
I can see that in the tumor.
Yeah.
You're fragmented.
Yeah.
However, heat shock is a transient phenomenon.
Okay.
They can recover within about 36, 48 hours. Okay. Right? So in other words, in that transient period, they're going to make altered O-glycan. They're going to make altered N-glycan also. Hopefully we can get that paper out by end of the year about this alteration of targeting on the O-N-glycan structure. That's how it can explain why cancer cells can make high nanos. Okay?
Right. I guess the other question here in terms of, have you considered looking at this model system you're looking at in terms of cancer cells that are chemo-resistant? Because that's a very, very serious problem in patients.
Okay, question over here.
From UPenn. I think you mentioned this, but I didn't talk about it. So I want to ask you about retrograde transportation of Golgi protein to ER. So did you see whether these play a role on the Golgi fragmentation you saw under the stress and those conditions?
Yes, I think that's a complicated issue. Because, for example, glycosyltransferase this is where I got into trouble. Cell biologists think that it's a COP2 mediated. We show that COP2 does not involve in the Golgi targeting of glycosyltransferases, but COP2 actually can affect the target. Okay, and that's where the targeting issue and the giantin defective, and so that's where it would affect the other component, will affect the Golgi morphology.
So when you see those CoL2 syntheses not in Golgi, are they transported into ER or they get stuck in the
Well, it's degraded by proteasome.
Oh,
yeah. So you culture cell long enough, you don't see you're going to have a decrease in the protein. Even though you have a gene expression, that's that point I want to get across. You measure the gene expression, you see it, but you don't see the structure. But only under the stress condition where you don't see it. All right?
Okay, let's thank the speaker one more time. Thank you very much.